So I didn't try this. Okay. So this is the title of the granted project indeed. So our idea was to use a gene therapy approach of overexpression of the wild-type allele in a mouse model of the disease. And so the question, as you can see here, was the one I just mentioned. The working hypothesis was, could we restore? So the working hypothesis was to, if we could restore the mutated gene function overexpressing the wild-type allele, and to do this, we injected in the brain by ICV an adeno-associated viral vector expressing the wild-type allele. So these are the methods. So this is the study timeline. So newborn mice were bilaterally intraventricularly injected with either GFP control construct or the one expressing the wild-type allele. And then after weaning, we start behavioral analysis, performing motor tests, and later on we perform functional studies on neuronal function in acute brain slices obtained from these mice. So this is just to test the efficacy of vector injection that is widespreadly diffuse in both hemispheres. And the mice were obtained by the courtesy of Katanev. He already characterized these mice, which are viable, have late onset hyperkinetic movement disorder, show hyperactivity, hyperlocomotion without epilepsy, and have already discussed for other models enlarged ventricles, and they show decreased number of neuronal precursor in the cortex. So our aim was to reproduce some of these dysfunctions, try to see whether the treatment could repristinate the original conditions of the wild-type animals. So the first test we performed was locomotion test in DVC cages. These are special cages organized with a 12 platform connected with electrodes to monitor for the presence of mice on it. So it was a way to monitor animal movement without disturbing the animal. I should say that this analysis was not easy. At the moment, I mean, early evidence described no gross differences among wild-type and mutated animals, but now we are changing the analysis, so these are data we got yesterday from the lab. So now we see increased activity, motor activity, locomotion during the day, only in males, and this seems to be corrected by the expression of the wild-type allele. As for the night activity, the situation is not so clear. Anyway, we have a reduction also during the night that becomes even stronger with the treatment. With females, I don't show the results because we are still trying to complete them. So we test again these mice for their movement, so the average speed, the distance traveled, and their willing to go in the center of the arena. And as you see, we could only monitor a reduced presence in the center of the arena for these mice. What is interesting is that we could revert the phenotype treating mice as described with the wild-type allele. Another motor test we took from Dr. Katanev's lab was this accelerated rotor rod test. And similarly to his results, we found that mutant mice perform better in the accelerated rotor rod test, especially after 3 days of training, and wild-type allele treatment restore the phenotype, here in red, of the wild-type mice. We then look at neuronal function. So we perform, as I mentioned before, electrophysiological recordings in neurons, cortical neurons of the fifth layer. And we look at specific modulation of the properties of these cells. What we found is a general increase of resting potential in the neurons from mutant mice that was not recovered upon mice treatment. This is the only features that was affected because we have no variations in other neuronal characteristics. So then we look at neuronal excitability, and we found no difference in wild type and mutants as concern firing frequency in response to injected current. Nevertheless, we look in more in detail to different characteristics. We observe some alterations. So we investigated this inward potassium current that is dependent on HCN channels that are induced by membrane hyperpolarization and by cyclic nucleotides. And what you can see from here, or from the graph on the right, is that wild type have higher amplitude of these potassium currents that are strongly reduced in mutant mice. And interestingly, in wild-type allele-treated animals, we reverse the phenotype. You can also see here. So the importance of this modulation is that these channels are important to receive the signals from the neuronal network in the cortical layer. So when they are blocked, as in the mutant, we have that the signal received are more prone to induce neuronal excitability, so increase their firing frequency. Another study was focused on, again, potassium currents induced by jerk channels activated by GABA-B receptors. And again, in these neurons, we observed that the activation of this current by baclofen induced the potassium current that were strongly reduced in the C215Y mice. And again, we could revert the phenotype in wild-type allele-treated animals. Again, you can appreciate from this graph. Thus indicating, again, that inhibitory neurotransmission was less efficient in these mutants, and so could be involved in increased neuronal firing, neuronal excitability. So this is just a scheme that describes how the GABA-B receptors are coupled to jerk currents, and how likely in this mutation we have reduced beta-gamma modulation of these currents. So To summarize, we characterize some of the functional effects of the C215Y mutation, inducing an increased resting potential, reduced hyperpolarization, reduced potassium currents that could explain in part some of the phenotype due to reduced or altered capability of these neurons to integrate cortical signals. And this kind of, let's say, gene therapy could be a promising tool. So, to conclude again, these data, I forgot to mention, are in accordance with the results shown this morning by Simone on C. elegans, and of course we would be happy to test further mutants with this experimental approach. So this is the list of colleagues that contribute to this work in Sapienza, in Istituto Superiore di Sanità, in the Italian Institute of Technology, and Professor Katanev, the colleagues that produced the AV construct, Professor Leuzzi and Serena Galosi as expert in the pathology, and Giorgio Rosati from Techniplus for DVC technology, and our colleagues in Rome at the CNR that help us to produce and genotype these mice. Thank you.